Hi everyone, my name is Natanya Keel. I'm from the McIntyre and Renee Labs at the University of Florida. Today I'm going to introduce you to SQANTI-reads, a tool we developed for evaluating the quality of replicated long-read RNA-Seq studies. And this project was done in close collaboration with the Canessa Lab. Over the course of this tutorial, I'll walk you through what SQANTI-reads does, how to run it, and how to interpret the results. So one of the first questions we need to ask is, why do we need quality control for long-read RNA-Seq? First, QC helps us identify the reads that map to annotated genes and transcripts versus reads that map to unannotated regions of the genome. We can also identify reads with technical artifacts, for example, degradation products or RT switching products. It also allows us to compare samples so we can evaluate bias in our experiments and identify low-quality samples. But the existing QC tools for long-read RNA-Seq have limitations. Most focus on basic metrics like read length and thread quality. What they don't capture is transcript structure, and that's really important for understanding long-read RNA-Seq data. So now that we know why QC is important, let's look at what makes SQANTI-reads different and useful. SQANTI-reads applies SQANTI-3 structural categories at the individual read level. We can also evaluate raw reads from multiple samples together in one run. It produces multi-sample summary files and visualizations, which makes it easier to compare across replicates. And finally, it helps identify potentially under-annotated genes and unannotated transcripts from the data. So on the right, you can see the basic workflow. Reads from different samples go through genome alignment with Minimap2, structural classification with SQANTI-3, then read and gene level metrics are calculated, and the output is a multi-sample QC report and summary files. Now we'll discuss the input files that are required for SQANTI-reads and the outputs it produces. We always start with two core inputs, a reference annotation in GTF or GFF3 format, and a design file that lists sample IDs, file accessions, and experimental factors, which I'll go over more in detail later in this tutorial. Then, in simple mode, you provide raw reads as FASTQ files or aligned reads in GTF format, along with the reference genome in FASTA format. In fast mode, which we recommend, you provide pre-computed SQANTI-3 QC output directories, which should include the classification and junction files. As output, SQANTI-reads produces a modified classification file with junction strings and junction hashes called reads_classification.txt, summary tables like gene counts, UJC counts, and read length distributions, annotation files, and QC plots, both in a default PDF and optionally in an interactive HTML report. Since SQANTI-reads builds on SQANTI-3, let's quickly go over the structural categories for those who are less familiar with them. Here we have the main categories. At the top is our reference transcript from the reference annotation. A full splice match, or FSM, is when the read matches all splice junctions of the reference. An incomplete splice match, or ISM, is missing junctions on either the 5' or 3' ends or both. Novel in catalog, or NIC, has a new combination of known splice sites, and novel not in catalog, or NNC, has at least one splice site that is not annotated. This is just a brief overview, and if you'd like more detail, this SQANTI-3 wiki is a great resource that explains all structural categories with examples. Now we're going to get into the features of SQANTI-reads. I will focus on introducing 4 key features and how we can apply them to real datasets. Feature 1. SQANTI-reads groups mapped reads based on their junction patterns into groups referred to as unique junction chains, or UJCs. The number of reads assigned to each UJC is recorded, and UJCs can be tracked across samples and conditions. UJCs are also classified as novel or known splicing patterns relative to the provided reference transcriptome. Feature 2. SQANTI-reads also identifies putative unannotated transcripts which are not present in the reference annotation. A novel UJC that is represented by a large number of reads indicates a novel splicing pattern that can be followed up. SQANTI-reads uses a customizable logic tree to fish out these high-confidence novel UJCs from the long-read data. Feature 3. SQANTI-reads quantifies the variation around annotated junctions by calculating the distance of each read donor or acceptor from the nearest annotated junction. We then calculate the mean, standard deviation, and coefficient of variation around each annotated junction and classify it as clean or messy based on the calculated coefficient of variation. Feature 4. SQANTI-reads produces several multi-sample summary files and visualizations that can be used to check sample qualities. These visualizations can be faceted by experimental factors like technology to evaluate different components of the experimental design. SQANTI-reads also performs a principal component analysis to identify which metrics drive sample variability. As I mentioned before, reads that share all internal junctions are grouped into a representative unique junction chain, or UJC, and the number of reads associated with that UJC is recorded. The way that we track these unique junction chains is by first generating a string containing the position of all junction starts and ends in every read. We then convert this string to a hash for computational efficiency. Using this hash, we can then quantify the number of reads assigned to each UJC and track these patterns across samples and conditions. The junction hash and string are provided in the reads classification.txt file so that each read can be linked to its hash. Now let's walk through an example. Here I'm showing an example set of reads mapped to a gene shown in gray and the corresponding annotated transcript in that gene shown in black. These reads are then assigned one of the SQANTI-3 structural categories, which we reviewed earlier in this tutorial. These reads are then grouped into UJCs, and the number of reads assigned to each UJC is recorded. In this example, we have a gene with a total of 104 reads and 5 UJCs. In general, we see that for high-quality datasets with a well-annotated reference transcriptome, 70 or more % of the reads represent an annotated splicing pattern. And in this experiment, we see that this is consistent across the 3 replicates. However, when we collapse reads to UJCs, we see that only a small proportion of UJCs have annotated splicing patterns, and the majority of UJCs have novel splicing patterns. This is because in general, we see that there are a small set of full splice match UJCs represented by a large number of reads, and there are many novel UJCs that are represented by few reads and in many cases only one read. So if we go back to our example, we can see that there's an FSM that represents 38% of the reads in the gene and an NNC that represents only 2% of the reads in the gene. However, there's another group of UJCs of interest, which are novel UJCs that are represented by many reads. To identify these novel UJCs of interest, we use a data-driven approach with a customizable logic tree to find them. First, we select genes with greater than 100 reads. Then we select all of the novel UJCs in these genes and identify those UJCs which represent more than 20% of the reads in the gene and have more than 80% of the maximum number of junctions observed in the gene. The reason that we care about the number of junctions in the UJC is to prevent us from selecting UJCs that are fragments of a longer transcript that could be degradation products. All of the thresholds set in this logic tree can be adjusted by the user to be more or less selective. We applied this logic tree to a long-read RNA-Seq benchmarking dataset of the WTC11 iPSC cell line and found 424 putative novel transcripts which are highlighted on the scatter plot. This scatter plot shows the percentage of max junctions in the gene versus percentage of total gene coverage, and each data point represents a unique junction chain. About 3/4 of these are novel in catalog, meaning that they represent a novel combination of annotated junctions, and the remainder are novel not in catalog, meaning that they have at least one unannotated junction. Now feature 4. To quantify variation around splice junctions, we use a metric known as the coefficient of variation, or CV, where we calculate the mean and standard deviation of the absolute distance of all read donors and acceptors to its nearest reference donor or acceptor. Based on the calculated CV, we then classify reference donors and acceptors into three categories. 1. Reference match, where all reads match the annotated donor or acceptor. 2. CV equals 0, where all of the reads, donors, or acceptors align with each other but do not match the annotated donor or acceptor position. Or 3. CV greater than 0, where there is some degree of variation around the annotated donor or acceptor. A larger CV corresponds to a greater degree of variation around the annotated donor or acceptor. So both reference match and CV equals 0 can be considered a clean splice site, and CV greater than 0 can be considered a messy splice site. This example comes from the LR-GAS benchmarking effort using the WCC11 human iPSC cell line that I mentioned earlier. We use triplicate transcriptome measurements from 3 different library types: cDNA PacBio, cDNA Oxford Nanopore, and direct RNA Oxford Nanopore, and all of them were processed through SQANTI-reads. Earlier I mentioned that SQANTI-reads can be run in simple mode or fast mode. Now, I want to go into more detail because this choice really affects how you can set up your analysis. So fast mode assumes that you've already run SQANTI3QC on each sample. The advantage here is that you can parallelize those runs using your system scheduler, for example, Slim, GNU Parallel, or whatever you prefer. This gives you much more control over mapping parameters, since you handle the pre-processing and alignment upstream. Another tip is to use the option skip off to skip off prediction when running SQANTI3QC. This will really help to speed up your individual runs. Now, in simple mode, SQANTI-reads does everything for you, starting with alignment using Minimap 2 by default. And then it runs SQANTI3QC sequentially across your samples. It's easier to set up and get started, but it can be much slower when you have many samples, so we typically don't recommend if you have more than 3 samples. Now let's take a closer look at the design file, since this is the key input that tells SQANTI-reads how to organize your samples. It's a standard comma-separated value file, or CSV. The sample ID column contains the names of your samples, and this is how they'll appear in your plots. The file accession column changes depending on the mode. In simple mode, it's the name of the FASTQ or GTF files. In fast mode, it's the name of the pre-computed SQANTI3 output directories. Then you can include any number of additional experimental factors. Here, for example, we've added library prep, replicate number, and platform. These extra columns let you facet or group the plots by experimental factors using the factor option. The design file basically acts as the blueprint for your analysis. It connects your raw or processed data with your experimental metadata and controls how the results are summarized and visualized. Here's an example of how you'd actually run a SQANTI-reads command. We start with the script sqanti-reads.py. The design flag points to the CSV design file that we just discussed. Input dir is the folder that contains either all your SQANTI3 outputs if you're running fast mode, or your FASTQ or GTF alignment files if you're running simple mode. Then we specify the reference, annotation, and genome files using the respective flags. dir sets the output folder, and prefix controls how the output files are named. With report, you can ask for PDF, HTML, or both as your report output, and here it's set to both. And finally, factor lets you tell SQANTI-reads which metadata column to use for grouping plots. In this case, library prep. I've also added a QR code here that links directly to the SQANTI-reads wiki. That's where you can find more details about how to set up the conda environment and other optional parameters that you might want to use. So here we can see one of the first SQANTI-reads outputs. It shows read distributions across structural categories. So here each bar is a sample, and then you can see that the bars are grouped by technology. The colors in the stacked bar plots represent the different structural categories. And so, for example, you can see in blue we have FSM, orange ISM, green NIC, and red NNC. In addition to the main structural categories, SQANTI-reads also report subcategories. Here you can see examples for full splice match and incomplete splice match. Each file, FSM counts or ISM counts, contains the numbers and proportions of reads in each subcategory. I won't go into detail on subcategories here, but if you're interested, the SQANTI3 wiki has full descriptions of how these subcategories are defined. Another important QC metric is read length. SQANTI-reads summarizes this in the file length summary.csv. This file reports the mean and median read length, the 25th and 75th percentiles, and the number and proportion of reads falling into bins like less than 1 KB, 1 to 2 KB, 2 to 3 KB, and greater than 3 KB. On the top plot here, we see stack bars showing the proportion of reads in each length bin. The violin plots at the bottom show the full distribution of read lengths per sample, highlighting not just the average but also the spread and outliers. Together, these plots help us quickly assess whether our read lengths are consistent across replicates and whether some technologies are producing systematically shorter or longer reads. SQANTI-reads also tracks common artifacts in the file errcounts.csv. These include RT switching, non-canonical junctions, and intra-priming. The bar plots here show the proportion of reads flagged for each type across samples and technologies. This is useful because artifacts can differ by platform and library prep, and seeing them broken down like this helps identify whether there might be a systematic issue. Now let's look at UJC metrics, which are summarized in the file ujccounts.csv. This file lists all the junction hashes detected in each sample, the number of reads assigned to each UJC, and the structural category of each UJC. In the top plot, you can see the total number of UJCs detected per sample, broken down by structural category. In the bottom plot, the same UJCs are shown, but stratified by how many reads support them. This ranges from singletons to highly supported UJCs. This gives us a sense of both the diversity of splicing patterns and the relative confidence in each UJC. Next are the gene level metrics summarized in genecounts.csv. Here, SQANTI-reads reports how many reads fall into each structural category within each gene, as well as how many UJCs are detected per gene. This allows us to see both the overall coverage of a gene and the different splicing patterns in each gene across samples. The plot on the right shows the number of genes detected in each sample, and the colors indicate how many genes are detected at different read depths. Junction metrics in SQANTI-reads are summarized in 3 main files. The first is junctioncounts.csv, which reports the number and proportion of junctions in each sample split into known versus novel and canonical versus non-canonical. This is shown in the top plot. The large blue bar represents known canonical junctions, while the smaller colored bars capture non-canonical and novel junctions. Next, CV.csv gives the coefficient of variation for each annotated donor and acceptor site, like we discussed earlier. This measures how consistently the reads align to the annotated donors and acceptors. Finally, CV donor or CV acceptor counts.csv aggregates those results into the CV categories that we defined earlier: ref match, CV equals 0, and CV greater than 0. The bottom plot shows this breakdown across samples and technologies. Annotation metrics are summarized in 2 key files. The first is geneclassification.csv. For genes that pass a user-defined coverage threshold using option gene expression, this file assigns each gene to a category: A. Well annotated with well-covered FSM, B. Well annotated with low coverage FSM, C. Under-annotated with a candidate transcript, and D. Under-annotated with no candidate transcripts. As you can see on the right, most genes fall into category A. The second file is putative under annotation.csv. This focuses on NIC and NNC UJCs, flagging potential novel transcripts that may not yet be in the reference annotation. These are identified using user-defined thresholds for coverage and junctions that we discussed in the flowchart earlier in this tutorial. Finally, SQANTI-reads produces a PCA based on all QC metrics. On the left, we see the PCA plot where samples cluster by sequencing technology. This separation shows that the QC metrics capture clear technology-specific differences. Here, we see that principal component 1 captures 56% of the variation across samples, and PC2 captures 35%. On the right, the bar plots show the top loadings for PC1 and PC2. These are the metrics that explain the most variation between samples. For example, separation along PC1 is driven by features like % novel non-canonical junctions and % NNC reads, which have the highest loadings. These aren't all the plots that SQANTI-reads generates. A full report can be found in the supplementary materials of the manuscript or in the example runs on the SQANTI-reads wiki. I hope this walkthrough gave you a clear overview of what SQANTI-reads can do and how to interpret sample quality from its output. So, in conclusion, SQANTI-reads is a powerful tool to compare different facets of replicated long-read RNA-Seq experiments and presents new methods for quantifying junction variation and identifying potential unannotated transcripts. Here I've provided the key references: the original SQANTI-3 paper, the long-read consortium benchmarking paper where the data come from, and the SQANTI-reads paper. I now want to especially thank Dr. Lauren McIntyre and Dr. Anna Canessa for their support and guidance throughout this project and the development of SQANTI-reads, and Dr. Carolina Monto for helping with the software implementation of SQANTI-reads and integrating it into the SQANTI-reads. I also want to acknowledge the McIntyre, Canessa, and Rennie Labs for their support. And finally, I'd like to thank all of the funding agencies who made this work possible. Thank you for listening to this tutorial of SQANTI-reads.